Welcome to another day of Mr. Mann's Anatomy and Physiology class. Hope you guys enjoy. Today we're going to talk about Unit 9, or the nervous system. We're going to have a couple of objectives here. We're going to learn the basic function of the nervous system, and we're also going to explain the structural and functional divisions within the nervous system to start. So as you see in this picture below, we have our sensory receptor, we have our sensory input, we have some integration that occurs within the brain and spinal cord, and then we have a motor output. And so we probably remember this from what we talked about previously within like homeostasis would be our efferent pathways and our the afferent pathways and the effector pathways. And then we had our control center here. So that's what you're going to be able to kind of see of how the nervous system plays a role within those pathways. So first, I'm going to zoom out just a little bit because we have some things over here. So our nervous system functions, we have a couple. So we have our sensory input, or what we call the afferent pathway. And the nervous system uses millions of sensory receptors to monitor the changes, both inside and outside the body. And we're going to collect that data like we talked about previously, and that's called sensory input. So anything that has to do with inside or outside the body to be able to look for changes within the environment of some sort is going to be a sensory input, whether it be pH levels, temperature, oxygen levels, or carbon dioxide levels, even touch. So then we have integration. So the nervous system processes that interpret sensory input, decides what should be done at each moment. That's when we have integration, or that control center. So we take their sensory input, we take it to the brain, and we try to figure out what do we need to do with this information. And so we take a lot of sensory input that we do nothing with because our brain goes, okay, we're good. It's not really that big a deal. Okay. Motor input. So the nervous system activates the effector organs, muscles, glands to cause a response, or what we call the motor output. That's going to be our efferent pathway. So now we have some divisions of the nervous system. So I'm going to zoom in a little bit. There we go. A little bit more. So some divisions, we have the central nervous system and the peripheral nervous system, or what we call the CNS and PNS. The central nervous system consists of the brain and spinal cord. And the CNS is going to integrate the control center and interprets the sensory input and dictates the motor output. So this is our control center, which is the central nervous system, or the CNS. The peripheral nervous system is going to be part of the nervous system outside of the CNS. So we have the brain and spinal cord, and then everything else outside of that is going to be our PNS, or peripheral nervous system. And that's going to be consistent of nerves, or what we call bundles of axons, that extend from the brain and spinal cord and the ganglia, or the collection of neuron cell bodies. And so we have spinal nerves and cranial nerves here, and the spinal nerves are going to carry impulse to and from the spinal cord, and the cranial nerves are going to carry impulses to and from the brain. And so we have two divisions of the PNS that are very, very important. We have the sensory, or the efferent division, and that's going to consist of nerve fibers or axons that convey nerve impulses to the CNS, and these receptors located throughout the body. And then our motor, or efferent divisions, and these are going to be transmits impulses from the CNS to the effector organs. So that makes sense. The sensory or afferent division is going to take it to the brain, and then the motor, so they can figure out what to do with it, and the motor and efferent is going to take it away to the organs to carry out the function. So we have two divisions of motor division. We have somatic and autonomic. So a somatic nervous system is composed of somatic nerves that conduct impulses from the CNS to skeletal muscles, and this is conscious control. So you pick up your Apple Pencil to write these notes is part of the somatic nervous system, while the autonomic nervous system consists of visceral motor nerve fibers that regulate activity of smooth muscle, cardiac muscle, glands, and this is what we call involuntary nervous system. So for instance, you breathing right now that you didn't think about that you were breathing until now that I told you is part of your autonomic nervous system. So we have two divisions of the autonomic nervous system. We have the sympathetic and parasympathetic, and you've heard these just in different terms, especially if you've taken psychology before. So our sympathetic division mobilizes the body systems during an activity, or what we call fight or flight. Okay? Then we have the parasympathetic division, is the housekeeping functions during rest. So this is going to conserve energy. This is going to help with food digestion, things like that. Now, we are going to talk about the neuroglia now. And this is going to support and maintain neurons, and the neuroglia is in the central nervous system. So we have our, zoom in just so that you can read this a little bit better, is the astrocytes, or what we call the star cell. And this is the most abundant glial cell. And this is going to support and brace neurons and anchor them to nearby capillaries for blood supply, oxygen, things like that. And it's going to mop up leaked potassium ions and recapture and recycle neurotransmitters, because we're releasing neurotransmitters, and then from there, they don't always get to the neurons. So we've got to recycle those so we can use them for later. Then we have some microglial cells, and this is going to monitor neurons and health and protective role. And special types is a macrophage, which phagocytizes microorganisms or debris. Okay, so phagocytes or macrophages are part of the immune system, and they're basically going to eat up microorganisms or debris that are harmful or could cause interruption to what we need these processes to happen. So we have epidermal cells, and this is going to be a wrapping garment. It's like cerebral spinal fluid they line the cerebral spinal fluid cavity, cerebral spinal fluid cavity, sorry I can't read, of the brain and spinal cord. And their cilia help circulate the cerebral spinal fluid, cushioning the brain and cord. I'll scroll down just a little bit for us. So now we have part D, the oligodendrocytes. And their cellular processes wrap tightly around the nerve fibers, producing an insulating covering, what we call a myelin sheath. And this is going to be able to help speed up our electrical impulses going down neurons, which we'll look at here in a little bit. So now we have the neuroglia in the PNS. So we have satellite cells that's going to surround the cell bodies in the PNS, and it's similar functions to the astrocytes in the CNS. And so if you need to know what the astrocytes do, they are going to hold them and anchor them to nearby capillaries and help mop up our neurotransmitters and potassium ions. Dealt with that electrical impulse down the cell. So now we have Schwann cells, and this is going to surround all nerve fibers in the PNS. They're going to form a myelin sheath around the PNS, and similar to the oligodendrocytes from the CNS. And so now we have some pictures for you on what's what. So here's our astrocytes, and this is our capillaries here that's helping anchor microglial cells, or epanchymal, our oligocytes, that sheath. And then within our PNS, we have our satellite cell here, and then we have our Schwann cells, and we can see we have little nodes of sheaths that we will see the nodes of Rendezvous. We'll talk about here in a minute. So now we have neurons. Okay, so neurons are also called nerve cells and are structural units in the nervous system. Now we have billions of highly specialized cells that conduct messages in form of nerve impulses, and we have some special characteristics of these. So they're going to have extreme longevity, and if we eat right and do the things we're supposed to do, then they can last a lifetime. We have neurons that are embryonic, and they have no ability to divide. Or if you remember from biology, we're stuck in G0 stage within the cells, within cell division. And the neurons have an exceptionally high metabolic rate and require a conscious and abundant supply of oxygen. And without that, we only survive for about a few minutes. So we have some neuron structures here. So we have the cell body. Within the cell body, we have the nuclei and ganglia. And the nuclei are clusters of cell bodies in the CNS, while the ganglia is clusters of cell bodies in the PNS. We have the neuron process. We have arm-like extension from the cell body. We have dendrites, short tapering branching extensions. They convey incoming messages towards the cell body. We have the axon. Zoom in just a little bit. It only extends from the cell body. It can be up to 3 to 4 feet in length, and they transmit nerve impulses away from the cell body. We have our nerve fiber, which is a long axon, a tract, which is the bundles of axons, and a nerve, which is the bundles of axons in the PNS, while the tract is in the CNS. So now we have the myelin sheath, and this is a whitish, fatty protein lipid, and it's segmented. And this is going to protect and electrically insulate fibers. So basically what's going to happen is going to increase the nerve impulses or the transmission speed within this insulation of these fibers and these fats and proteins. And so it's going to conduct nerve impulses rapidly, and non-myelinated sheaths conduct nerve impulses slowly. So we see here, we have a picture here. So these are myelin sheaths, or what we would call Schwann cells. Okay, and any unwrapped portion of the myelin sheath or Schwann cells is going to be what we call the nodes rendezvous. So basically what's going to happen is it means nodes of jumping. It's French. So we have our cell body, we have our axon. Impulses are coming through these dendrites here. Whoop, forgot to label dendrites. There it is. And they're going to travel down. Well, they're going to travel down through what we call these nodes of rendezvous. So they're going to literally jump, or nodes of jumping of this impulse. As they jump, they increase the speed, so they're not going all the way down this entire axon to where we get to the axon terminal here. And that's why they speed up the electrical impulse. Now, myelination in the PNS, myelin sheaths in the PNS are formed by Schwann cells, and they wraps around the axon like a jelly roll. And the rons and rendezvous are gaps in between the Schwann cells, which we've already talked about within the picture. Okay. Then myelination in the CNS, the myelin sheaths in the CNS are formed by oligodendrocytes, oligodendrocytes. Okay. So we can see here this wrapping of this fatty protein. We have the axon here, and then this is our Schwann cells, and then on the either side would be our nodes of rendezvous. Now some classifications of the neurons. Structural classification, we have multipolar, and it's the most common type, 99% of neurons. They have three or more processes, one axon and the rest are dendrites. We have bipolar, which is going to be found in retinas of the eyes and the olfactory mucosa of the nose, and it's two processes, one axon and one dendrite. And then we have unipolar. It's a single short process that emerges from the cell body and divides into two. So here we have a multipolar, so we see multiple dendrites here. 1, 2, 3, 4, 5, 6, 7, 8. And then we have one axon, and then this would be our axon terminal here. So this is going to be a multipolar. While this is a bipolar, we have one cell body, one dendrite, and it's only going to go within one direction. As we see there, we have two processes, one axon and one dendrite. While unipolar is here, we have literally just one axon, and it's short, and we have axon terminals on both ends, and that's unipolar. So some functional classifications. We have sensory and afferent neurons, and these would be from sensory receptors to the CNS. Almost all are unipolar, and the process can be very long with ganglion close to the cord. We have our motor and efferent, which is from the CNS to the effector organs, and these are all multipolar. And then we have interneurons, which is 99% of neurons, and they lie between the sensory and motor neurons, and they shuttle signals from the CNS pathways during integration. So that's going to be it for notes, guys. I hope you guys learned a lot, and I will see you on Tuesday or Wednesday, depending on either your A or B.